Hello everyone, very warm welcome to India's largest online learning platform ITLS Academy. Our today's topic is undifferentiated stage of a mouse embryonic stem cell. This is continuation part of embryonic stem cell that we have discussed in our previous lecture. In this particular video, we will see undifferentiated state. So let's start. Undifferentiated state of a mouse embryonic stem cell. The very first point is the undifferentiated state of the embryonic stem cell is characterized by specific cell markers that have helped scientists better understand how embryonic stem cells under the right culture conditions replicate for hundreds of population doublings and do not differentiate. A true stem cell is capable of maintaining itself in a self-renewing undifferentiated state indefinitely. Next point is the cell cycle of the embryonic stem cells also seems to play a role in preventing differentiation. From studies of these various signaling pathways, it is very clear that many factors must be balanced in a particular way for embryonic stem cell to remain in a self-renewing state. Next point, stem cells are referred to as undifferentiated cells because they have not yet committed to a developmental path that will form a specific tissue or organ. The process of changing into a specific cell type is known as differentiation. If we talk about this diagram, this picture is showing culture of mouse embryonic stem cell. We have seen a major difference in embryonic stem cell and embryonic germ cell. The culturing procedure of these cells are given in this particular picture. Here another embryonic carcinoma cells are also given, and we will see culturing procedure of carcinoma cells also. So first, if we talk about the embryonic stem cells culture, you can see this is blastocyst, this is inner cell mass, This is mouse blastocyst, as we are studying each and everything in the case of mouse. So for the culture, these inner cell mass cells are taken in this particular petri dish or culture plate. These inner cell mass cells are further transferred, or a transfer of stem cells to mouse blastocyst has occurred. These are normal blastocyst cells are formed. Next comes embryonic germ cells. Now we are going to see the culturing procedure for the embryonic germ cells. These are mouse embryo cells. Here comes primordial germ cells. These primordial germ cells are taken for culture. In the culture media, you can see here these are normal blastocysts. Next comes embryonic carcinoma cells. These are taken from testicular teratocarcinoma. These are taken in culture media. And again you can see here normal blastocyst. So this is all about how culturing of embryonic stem cell, embryonic germ cell and embryonic carcinoma cells is done. Here a question arises: can a mouse embryonic stem cell be directed to differentiate into a particular cell type in vitro? Is it possible? So very first point for the answer is, one goal for embryonic stem cell research is the development of specialized cells such as neurons, heart muscle cells, endothelial cells of blood vessels, and insulin secreting cells similar to those found in the pancreas. Differentiation of embryonic stem cells in culture. When removed from the factors that maintain them as stem cells, embryonic stem cells will differentiate and, under appropriate conditions, generate progeny consisting of derivatives of three embryonic germ layers, and these three embryonic germ layers are mesoderm, ectoderm and endoderm. Next point is, by far the most common approach to directing differentiation is to change the growth conditions of the embryonic stem cells in specific ways, such as by adding growth factors to the culture medium or changing the chemical composition. During development, the visceral endoderm, or we can call it as primitive endoderm, and epiblast, as we know epiblast is also known as inner cell mass, are derived from the inner cell mass of the blastocyst. Third point is cell-cell interactions are critical to normal embryonic development. Interaction between two cells becomes a very critical or plays a very critical role for the normal embryonic development. Here in this particular diagram, you can see how these three germ layers are formed. These are stages. This is zygote formed after the fusion of egg and sperm. This zygote undergoes the process of division. This single cell forms two cells after division, and after two cells, these two cells give rise to four-celled embryo, next 8 cells, and when it comes to 16 cell, it forms compact morula stage. This morula stage further forms early blastocyst. This early blastocyst undergo differentiation and forms mid-blastocyst. This mid-blastocyst further forms late blastocyst. This late blastocyst then forms epiblast, and this epiblast give rise to mesoderm, endoderm and ectoderm, that is all three germ layers. Here you can see this is epiblast. This is taken for derivation in cultures medium. Here embryonic stem cells, these go differentiation, and you can see after differentiation these forms a complete mouse body. If we talk about totipotent blastomere, this is blastocyst stage. Blastocyst is made up of these small cells. These cells are known as blastomeres. So if we talk about this blastomere, so this is totipotent. This every cell can give rise to a complete embryo. That's why it is known as totipotent. This totipotent blastomere forms trophoectoderm and inner cell mass. This trophoectoderm formed by the activity of CDX2. I will write here. This is for the trophoectoderm. This is CDX2. This is a signaling pathway. GATA3, GATA, and EOMs, EOMES. These are for the trophoectoderm cells, and signaling pathway for inner cell mass includes OCT4, NANOG, I am writing because the visibility of this part is low. And next for the inner cell mass is GATA 5. Guys, you can take a screenshot of these signaling components. I hope you all taken a screenshot. So if we talk about the inner cell mass differentiation, this forms pluripotent epiblast. The main signaling pathway for this epiblast is nanog. I am again writing here. This is nanog. And the other is primitive endoderm. The pathways for signaling pathway for primitive endoderms are GATA 5, SOX 17, SOX, SOX 17, GATA 4, and SOX 7. These signaling pathways help in the formation of primitive endoderm. This whole signaling pathway is part of cell biology. For reference, I have taken this diagram. You can see how signaling pathways help in the formation of each and everything in the body. So guys, again you can take a screenshot of these signaling pathways if the visibility is low. Otherwise, you can also understand for this diagram. Another way to direct differentiation of embryonic stem cells is to introduce foreign genes into the cells via transfection or other methods. Next point is the approach appears to be a precise way of regulating embryonic stem cells differentiation, but it will work only if it is possible to identify which gene must be active at which particular stage of differentiation. This is a very crucial step in differentiation and regulation of embryonic stem cell. The result of these strategies is to add an active gene to the embryonic stem cell genome, which then triggers the cells to differentiate along a particular pathway. Another approach to generate mouse embryonic stem cells uses cloning technology. The resultant pluripotent cell would be immunologically compatible because it would be genetically identical to the donor cell. Next point, all of the techniques just described are still highly experimental. It is not possible to explain how the directed differentiation occurs. However, no one knows how or when gene expression is changed, what signal transduction systems are triggered, or what cell-cell interactions must occur to convert undifferentiated embryonic stem cells into precursor cells and finally into differentiated cells. Embryonic stem cells have been shown to differentiate into a variety of cell types. For example, mouse embryonic stem cells can be directed in vitro to yield vascular structures, neurons that release dopamine and serotonin, and endocrine pancreatic islet cell. In all the three cases that we have discussed, proliferating undifferentiated mouse embryonic stem cells provide the starting material and functional differentiated cells were the result. In addition, when directed to differentiate, embryonic stem cells aggregate a change in their 3D environment that presumably allowed some of the cell-cell interactions to occur in vitro that would occur in vivo during normal embryonic development. And all three studies demonstrated that the resulting differentiated cells functions as their in vivo counterparts do. These two criteria, that is, demonstrating that a single cell can give rise to multiple cell types and the functional properties of the differentiated cells form the basis of an acid test for all claims of directed differentiation of either embryonic stem cells or adult stem cells. Very few experiments meet these criteria, which too often makes it impossible to assess whether a differentiated cell type resulted from the experimental manipulation that was reported. Here you can see in vitro handling drop method for culturing of these pluripotent embryonic stem cells is given. If we talk about the procedure, so these are mouse embryonic stem cells. These are at day 0. Embryoid body formation occurs after 0 day. After 1 to 3 days, there is hanging drop. You can see a structure of these hanging drops. Differentiation of these hanging drops occurs after day 4 to 17. There is formation of all three germ layers, that is ectoderm, mesoderm and endoderm. This is in vitro handling drop method for mouse embryonic stem cells. Here you can see how all these cells are formed. Here step by step with a signaling pathway all the things are given. You can see this is morula state, this is morula. Here the signaling pathway includes OCT3-4 and CDX2. In the previous diagram it was not very clear, but now it is very clear. You can easily understand these two signaling are going on. If the signaling pathway is OCT3 or OCT4, then it will form inner cell mass. And if the signaling pathway is CDX2, then it will form trophoectoderm. You can see this formed structure is known as early blastocyst. This blastocyst have a blastocyst cavity. This early blastocyst also undergo some signaling pathway. This is nanog pathway. This nanog form epiblast. And here another signaling pathway, that is GATA6, also occurs, and this forms primitive endoderm. This primitive endoderm further differentiates and forms visceral endoderm and a parietal endoderm, while this epiblast undergo differentiation and forms primitive ectoderm. This trophoectoderm directly undergo extra-embryonic ectoderm differentiation. So you can see here this primitive endoderm formed after the signaling pathway GATA6 forms visceral and parietal endoderm, while this trophoectoderm formed after CDX2 signaling forms extra embryonic ectoderm, while this epiblast forms primitive ectoderm. Further, this primitive ectoderm forms germ cell lineage and also forms somatic cell lineage. This somatic cell lineage forms ectoderm, mesoderm and endoderm, while germ cell lineage will form gametes, that is eggs and sperm. So thank you guys for watching this session. Follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. For latest videos and updates, subscribe to our YouTube channel, that is ITLS Academy, and you can also get it on Google Play Store. Thank you very much guys for watching this session. If you like this video, then please like it, also share with your friends and subscribe ITLS Academy. For any help, you can call us on our helpline number 7080833450. Thank you very much guys for watching this session.